Hello friends, we will continue with nitrogen metabolism and this is the third part. The question is, what role do the flavonoids play in the nodule formation? So this I have discussed in the last lecture also. I am again discussing this with you that the leguminous plant released some kind of chemicals. These chemicals are isoflavonoids and these chemicals attracts the bacteria towards itself. So these isoflavonoids, they are the plant phenolics which are released by the host plant. These host plant are leguminous plants. Okay, so I write here that isoflavonoids or flavonoids, they are plant phenolics. They are plant phenolics and they stimulate, which first attract the bacteria towards the host plant. After that, it stimulates the nodule formation. So nodule is not formed on the plant all the time. It is formed only when the bacteria come in association with the host plant, right? And also stimulates the nodule formation. Okay, so they are released by the roots also. Okay, they are released by the roots also, and because of this they attract the bacteria towards itself. Okay, I'm drawing the bacteria with blue color. They attract the bacteria towards itself. Okay, when there is association between the bacteria and the host plant, then the further step is the nodulation or the nodule formation. Okay, some genes will be activated, then there will be the nodule formation inside which the bacteria will inhabit and will fix the nitrogen. Okay, next is, what are nod factors? So see, what are nod factors? So you will see my previous videos, I have discussed all this in detail. So first of all, I would like to explain you a process in little detail. These nod factors, what is the chemical nature of these nod factors? These nod factors, they are lipo lipokyto oligosaccharides. These nod factors, they are released by the rhizobium bacteria. Okay, so they are released by rhizobia. Where are they released by the rhizobia or rhizobium bacteria? So they are released by rhizobium bacteria. I write the rhizobium. Where? In the rhizosphere. What is rhizosphere? Rhizosphere is the area near the roots or in the vicinity of the roots. Okay, they are released by rhizobia near the rhizosphere. Okay, and these nod factors, when enter the plants, when enter the host plant, what will happen? It causes some change in the host plant. Okay, these nod factors, when they are released in the rhizosphere, when these nod factors enter the host plants, they causes some change in growth and metabolism of the host root. So what I said that whenever this rhizobium bacteria come in contact with the host root, several steps are seen one after the other. Okay, these bacteria, when they release the nod factor in the rhizosphere. So this region is a rhizosphere. Okay, this region here, this is a rhizosphere. When these bacteria release this nod factor in the rhizosphere, and when these nod factors enters the host plant, it causes the change in growth and metabolism of the host root. After which, what we see that there is the formation of nodule. So what are these nod factors doing? They are showing some change in growth and metabolism of host root, as a result of which nodule is formed. Clear? So ultimately their main role is formation of nodule on the host root. How? So at first bacteria become attracted to the host root. They release nod factor. These nod factor, when they enter the host plant, they causes growth and metabolism in the host root, and as a result of which nodule is formed. Up to here, is this clear? Okay. So here, what are lectins? Lectins you must have studied in several chapters. Here, what does it mean? That first thing you must remember, that the ability of nitrogen fixing bacteria, which is rhizobia, they form the symbiotic relationship with legumes, with and this symbiotic relationship is characterized by high degree of selectivity. So here I write here these the association, the symbiotic association between the leguminous plant and rhizobium is very selective in nature. For example, this rhizobia will only form the symbiotic association with pea plants and with no other legume species, such as they will not show the association between the soybean plant, the jack beans. They only will show symbiotic association with pea plants only. Okay, so these lectins, they are carbohydrate molecules. These lectins are carbohydrate molecules released by the host plant onto their surface. Okay, released by host plants on their cell surface. Okay, they are carbohydrate binding proteins. So these are basic ideas we all have. Okay, so when the nod factors are released in the rhizosphere, these nod factors, which are lipo, keto, oligo, polysaccharide in nature, they will bind with the lectin molecules. If the binding is specific, then only the further process will start. Otherwise, the process will stop there only. Okay, so when the binding is specific, then only there will be the further process that will start, otherwise the process will stop there and there only. So what do? If I write the function of lectin, they have two functions in nitrogen metabolism or nitrogen fixation. First is they help in determining the specificity, that there is a correct association between the rhizobium and the pea plant or not. So determining the specificity between the nitrogen fixing bacteria and their host plant. Secondly, they help in hydrolysis of lipocyto oligopolysaccharide of the bacteria. Okay. So again I repeat, what do lectins do? These lectins at first they are carbohydrate molecules. Sorry, they are carbohydrate binding proteins. So I am correcting this one. They are carbohydrate binding proteins. Okay, they are released by the host plant on their cell surface and they helps in selective association between the leguminous plant and the rhizobium. That is, rhizobium shows symbiotic association with only pea plants and not with other leguminous plants. So due to presence of this lectin, rhizobium can identify whether this is the correct plant or not. Okay, so what do? They helps in determining the specific association between the nitrogenous bacteria and the plant host, and also they helps in hydrolysis of lipocyto oligosaccharide of bacteria. Clear? So what is infection thread? So these infection thread, they are internal tubular extension. They are internal tubular extension. As you can see here, the one in the brown colored part, these are the thread where the bacteria, inside which the bacteria are present. Okay, these thread will branch and reach to different host cells. So these are host cells. Okay, these are the roots, and this is root hair, and these are actually the root cells, and this is the brown colored part which you can see here. It is the infection thread. Okay, and these are bacteria. So these are tubular extension of plasma membrane of root hairs. Okay, through which the rhizobium bacteria will enter the cortical region. So this region is a cortex. So how are rhizobium entering into the cortical cells? How they are released in the cortical cells? Through this infection thread. Okay, so through infection thread, these rhizobia are released or reaches, these rhizobia, they reaches the cortical cells. Clear how these infection threads are formed? So many Golgi vesicles and tubules, they fuse together to form the tubular extension. These tubular extension have the bacteria, and these bacteria through the infection thread they reaches the cortical cells. Okay, where they will form the nodule. So see here, whenever these bacteria reaches the cortical cells, what will happen? That these bacteria will induce cell division. Okay, these bacteria will induce cell division. Okay, now this cell division, what will happen? It will induce cell division in nodule primordium. So what is nodule primordium here? Here, where the cells, where the bacterial cells are accumulated, this region will become nodule primordium because this is now going to form the nodule. Okay, so after the bacteria accumulates in a cell, they will induce the cell division in that region, and this region is nodule primordium. Okay, so see here, these are bacteria, okay, which where they have infected the central zone. Okay, next is after differentiation, we will find that they develop into circular bacteroids. What are circular bacteroids? That means they have formed the membrane around themselves. Okay, previously they were rod shaped, previously they were not circular, they were rod shaped. Okay, after differentiation, they form the circular bacteroids. Okay, they form circular bacteroids. When they entered previously, they were rod shaped. So, what is nodule primordium? If I write it in definition form, what will you mean by that? These nodule primordium here, the rhizobia, infected cortical cells that de-differentiate, means they will all have become permanent cells, okay, that de-differentiate and start dividing again. Okay, these cells are known as nodule primordium. So in a simple language we can say that wherever the bacterial cells will accumulate, these cells become nodule primordium where the bacteria induce more and more cell division and lead to the formation of nodule. Okay, nodule is the region where the bacteria will fix the nitrogen. Clear? So that's it for today. We will continue in the next lecture. Thank you.